Morning, Ma. Um, we have some participants in-house and then we have some other participants online. Let's welcome Dr. Monsurat Gbadamosi, and she'll be talking to us about ALS and Drosophila research. Thank you so much, Ma.
Okay. I don't know. Okay. Good morning, and thank you very much for inviting me to this workshop. And I would like to tell you something about TDP43 in Drosophila and what we have used it to do in ALS research. Um, I'm sure Dr. Borati has told you all about TDP43. He's the PDP43 expert. But I'm going to tell you what we have done with it in Drosophila. ALS is a neurodegenerative disease. It's progressive. Usually, people die 3 to 5 years after diagnosis because it leads to the majorly degeneration of the motor neurons. So, but for us, we were interested in what happens prior to this event. Why do motor neurons die? The hallmark is the death of the motor neurons. Before that happens, what has happened in the body before it got to that motor neuron death? So that is why we decided to take some steps back, because many models uh, follow the TDP43 aggregation model, because what you see, the hallmark of ALS is um, TDP43 aggregation in the brains of ALS patients. So for us, we took a step back to see what happens to the neurons before they die. And for us, we think the synapses will fail first before the motor neuron die. So what we did was to check the synapse first. So let's get into it. But before I go deep into that synaptic architecture that I'm talking about, that we think failed before the neurons die, I'd like to quickly tell you why we think Drosophila is important, is ideal to use for this type of early neuronal study. Well, the usual one is that, Drosophila is fast. It has just 10 days of life cycle. You could do a lot of experiment in a little bit of space, and comparatively, it is cheaper to run experiment in flies than in mouse. So, but what is important, very important to us, is the conservation of the genes in Drosophila compared to humans, because Drosophila has just four chromosome, and majorly most of the genes are on the second and third chromosome, and in this he is able to have about 70% of the disease causing genes in humans have a functional autolog in Drosophila. So that means it is very, very, the genome of the fly is very, very compact. He has not a lot of waste compared to the human beings that has 22 pairs of chromosome, and then the fly with just four is able to recapitulate 70% of the disease causing genes in human to have a functional autolog in the fly. So that is very, very fantastic. And this hallmark protein of ALS, which is TDP43. TDP43 is what is called in human. In fly, we call it TBPH, which is TDP43 is star DNA binding protein 43 kilodalton, and TBPH is star DNA binding protein homolog, which is TBPH. So in this talk, I might use TDP43, I might say TBPH. I'm saying exactly the same protein, just that one is in fly and the other is in humans. So because TDP43 pathology has also been found to be conserved in flies and in humans. So Drosophila is very ideal to use for this kind of a study. And also we have molecular tools that we can use to do genetic manipulation. The most famous and versatile of them all is the US GAL4 system, which I personally love because it's easy to use and it makes your life quite easier when you want to do genetic studies. So for Drosophila, and also the functional readout of all these genetic experiments is pretty easy to see. When you're using Drosophila, you don't have to struggle a lot to see. We have behavioral assay, you can do NMJ. In fact, the NMJ of the third instar larva of Drosophila is an excellent model to study the um, synaptic architecture, because you can easily get to it. You can dissect, stain, and see a lot of stuff gross. So it's Drosophila is very ideal for studying this kind of early neuronal failure. And as I've said, TDP43 is the hallmark of the disease, because the protein is the most abundant pathological protein found in ALS, both sporadic and familiar. And usually it is the problem of the levels of TDP43 that is the basis of the models, various animal models of ALS that is available for us to use for research, because normally the protein is in the nucleus. It's supposed to be localized to the nucleus, an RNA binding protein. But when it is now mislocalized to the cytoplasm, is where it forms those aggregate that is later seen in the brains of these ALS patients. So the level of this protein has to be tightly regulated, because if it is overexpressed forming those aggregates, you're going to have problems of overexpression, which can also show and lead to symptoms of ALS. And if it is not present at all, because it's not in the nucleus anymore, and the function is supposed to be doing in the nucleus is lost, it will be loss of function model of TDP43, and we are also still going to have ALS being modeled. So this protein needs to be tightly regulated for the neurons to be healthy. Okay, now that's briefly about TDP43. Now, the Drosophila TDP43 model itself. This model that I'm going to talk a lot about today was created by figuring it out 2009. Well, that's my PhD boss. It was this fly is a loss of function model of TDP43. What that means is just that TDP43, the gene that is responsible for the production of TDP43 protein in the flies, has been completely knocked out. So the flies do not have TDP43 at all. So it's a loss of function model. So this model has been extensively characterized. It has reduced lifespan, locomotion problems, synaptic problems, and regulation of key synaptic proteins like syntaxin, Futsch. You have problems with synaptic vesicle recycling. The model has been extensively characterized and studied. And we know that it's also we know that this TDP43 fly model is useful to do ALS research, because most of the symptoms of ALS that we see in human beings, this fly recapitulates quite a lot of them. And when you try to do a rescue experiment using human TDP43, remember I said this is a loss of function model, meaning that the flies do not have any TDP43 at all. If you try to put human TDP43 into these flies, it rescued the symptoms that you see, the phenotypes. You rescue them using either human TDP43 or Drosophila TDP43. So this further validates the model for studying ALS mechanism, and it's also demonstrated that there is still functional mechanism. The mechanism of functionality of TDP43 is conserved between Drosophila and human beings. So we can use Drosophila to study ALS in human beings. These videos are just to show you, the phenotype that you see when you don't have TDP43 at all. Is the video playing? Okay, that's the
Yes.
Y type. You can see it's working pretty fast, pretty good. No locomotion problem, no coordination problem. And the one in the middle that I'm just playing now, you see, he has problem coordinating his movement. He cannot really walk. Can we see the difference?
Yes.
Now that is TDP43 mutant. It doesn't have TDP43 at all. So we can see that he has lost his ability. Look, he has a lot of locomotion defects, and this is also right down to the genetic and molecular level. And this is when you put human TDP43 back into that fly. You see?
Yeah, we
So
can
it is
see.
working.
Yes.
So yes, this for the, I just wanted to give a visual effect for this talk that I've been saying, that it rescued that. We can see this pretty clearly from the videos. So that's just, by the way, to show us that it's not just talk, it's working. You can see it, feel, not just molecularly or genetically, you can also see it physically, phenotypically, you can see it, and you are able to differentiate what is going on there. Okay, now my slide does not want to move. Okay. Okay, back to the model that I'm talking about. What happened was that, I told you that we were interested in understanding what was happening before the motor neurons die, before that hallmark showed up, the death of the motor neurons. So, because we have characterized the TBPH mutant a lot, we understand and we have seen that quite a number of key synaptic proteins were downregulated in the TBPH mutant. Proteins like syntaxin, DLG, Futsch, they have a direct physical relationship with TDP43 because it binds their messenger RNA. The mRNA is their direct targets of TDP43. So those ones are pretty understandable. But some of them, like let's say synapsin, CSP, these are uh, synaptic proteins. They were downregulated in TBPH mutant, but they are not direct physical target of the TDP43. So their mRNA is not direct target, because remember, TDP43 is an RNA binding proteins to, it works in a lot of ways: splicing, translation, transportation, quite a lot of functions, which is why the TDP43 protein is very, very important in the development of the whole animal. So you see, now we know that these proteins, the synaptic proteins that were there and downregulated in TBPH mutant, some of them are involved in vesicle recycling. They are vesicular proteins. So in order to probe this further, we decided to check the synaptic vesicle recycling system in the fly, to see what the role of TDP43 is, because some of the synaptic proteins that were downregulated were proteins of synaptic vesicles. So yeah, what we did was to get FM143. You see, this FM143 is just a dye that can be incorporated into the synapse, can be incorporated via the plasma membrane into the neuronal membrane this time around. Gets into the neural membrane, gets into the vesicles. It could also get through the process of endocytosis. It can also get out through the process of exocytosis. All this is you can do this by stimulation using potassium chloride. I have the protocol somewhere which I got from Vestrikin. That's one of our collaborators. Vestrikin in 2008 published this protocol, and it's wonderful because if you follow the protocol, it works quite well and it's easy to use. It doesn't have a lot of big, or let's say you don't need a lot of stuff to do it, but you do need a live microscope though, because all of this is live imaging. You need to fix, not necessarily it's live, you're not fixing the tissue, but you're just washing and stimulating it. There's a protocol, I can share it later if anyone is interested in seeing it. But just to summarize what happened when we did this, we tried to see how did we check the synaptic vesicle recycling. We did it trying to see what happens in the synaptic endocytic cycle, synaptic cycle, which consists of both endocytosis and exocytosis. Endocytosis is when we load in the dye, we term it that loading. Exocytosis is when the dye is being taken out of the vesicles into the synaptic cleft. So that is exocytosis, and we term that as unloading. So when you load in the dye, during the experiment, when you load in the dye, what you're trying to do is endocytosis. Then you stimulate again, you unload the dye. That is exocytosis. That is one complete synaptic vesicle cycle. So when we did this with the mutant, we found out that the mutant had problems loading, which translates to endocytosis. They also have problems unloading the dye that they have loaded. So that means they have problems also in exocytosis. So which is in line with what we suspected, the our hypothesis right from the beginning, because we have some synaptic proteins being downregulated. This is in line with it, that the mutants have problems in synaptic vesicle recycling, because this process is what is very important in synaptic transmission. So now that we know that TBPH is important, because if now that TBP, the mutants have problems loading and unloading, endocytosis, they have problems with it. What we did was to express TDP43. Remember, they don't have TDP43. So now we did a rescue experiment, putting TDP43 and see if they can do the endo and exocytosis. And yes, expressing TBPH in their neurons was able to rescue this process. So yeah, which is a validation experiment that confirms that TDP43 is involved in vesicle recycling. Okay, now that we know that TDP43 is involved in vesicle recycling, how is he able to do that? Because it's an RNA binding protein. How does he do that at the synapse? So we look into RAB GTPases. You see, these RAB proteins are documented regulators of synaptic not are regulators of vesicle recycling. So we try to check RAB GTPase 4 and 5. There are a lot of these RABs, but we stuck to two, RAB4 and RAB5. These RAB proteins, what we did with them was to express RAB in the neurons of these flies and see what happens. Remember, the TDP43 mutant flies, they have problems of locomotion, their neuromuscular junction, the branches are reduced, the buttons are misshaped. They have a lot of problems there. So when we express the RAB4 and RAB5 in the neurons of this fly, RAB4, we have recovery of the NMJ branches. Okay, yeah, this is RAB4, this is RAB5, this is wild type the control, and this is the mutant. If you see the branching, it's pretty obvious. You can see it here. RAB4 is doing something that RAB5 is not doing to the mutant. So, and then this is not just genetically or structurally, because the larva can also work better. This is it here. This is RAB4, this RAB5, this is the mutant. This is neuronal driver, and this is motor neuron driver, D42, and this is LRV, is a pan neuronal driver. It's expressed in all neurons. So what this says is RAB4 is a regulator that is involved in fast recycling vesicles. That's his area of that's what RAB4 does. But RAB5 is involved in like late endosome vesicles that are going to the late endosome. This is all about the endosomal cycle in the flies. But what I want us to take from this, the goal of this experiment was to check how specific this action of RAB4 is with TDP43, because both RAB4 and RAB5, they are endosomal proteins. They are RAB proteins that they are GTPases that are involved in synaptic vesicles. So we needed to know if this action of recovery that we're seeing with TDP43 is specific to RAB4, or RAB5 could also do that. So, but based on the result, RAB5 was not able to recover motility or the synaptic architecture either. So now we know that TDP43 has something to do with synaptic vesicle recycling, because if it's not that, the mutants have problems with their synaptic vesicle recycling. And we also know that RAB4, which is a GTPase, can rescue some of these defects that we see in the synaptic vesicle recycling, because RAB5 could not do that. So this action that we see is specific to RAB4. Okay, so we are taking on RAB4 now because that's the positive result. So we go ahead further to check what is happening at the neuromuscular junction of these RAB4 rescued flies. We checked the pre and post synaptic organization. For the pre synaptic one, we stained for Futsch. Why did we choose Futsch? We know that Futsch is a microtubule associated protein that is responsible, that works with the architecture, is involved in maintaining and stabilizing the synaptic architecture during division and growth. So Futsch is very important, and it's also a direct target, is a direct target for TDP43. So when we checked Futsch, the level of Futsch at the neuromuscular junction of these flies, here, this is the mutant, this is wild type, and this is RAB4 rescue. See, at the terminal buttons, Futsch is missing in the mutant, which is what is expected. This here, you can see Futsch here at the terminal buttons of the Y type, and RAB4 puts Futsch back into it. That's what we checked, which is interesting result if you ask me, because Futsch and RAB4 have not been connected before. They are different different proteins that does different things. What is connecting them now is TDP43. So at the postsynaptic, we tested the we stained for the clusters of glutamate receptors and for the scaffold protein DLG. Both of them were also recovered were recovered when we overexpress RAB4, which is interesting. But what we could get from here is that RAB4, that is regulating endosomal recycling, if you overexpress RAB4 in this mutant, it is sufficient to recover the presynaptic terminal and reconnect it with the postsynaptic terminal to improve synaptic transmission, which is interesting because now we have RAB4 and we have Futsch, which are doing this recovery synergistically outside of TDP43, because these flies do not have TDP43. Okay, so to validate this experiment, it's not just in flies. What we did was to validate RAB4 in ALS patient motor neurons. The first step we did was to get the brains of this lab actually that has this Drosophila mutant. We checked the mRNA using using qPCR, and then the level of the transcript was significantly lower in the mutants compared to the wild type. And then we did something called RNA-IP, where we use flies that, because what this is trying to do is to see, now that we know that the mRNA of RAB4 is significantly lower in the mutant compared to the wild type flies, what we want to know is this action, is RAB4 having a direct physical relationship, is the mRNA binding to TDP43. So that's why we did the RNA-IP, And what we did, we used a fly that has um that is expressing the normal TBPH, normal TDP43. So white eye fly, fine. And then the second group is the fly that has is expressing a TDP43 that has a 2-point mutation in its RNA binding domain. So it has TDP43, but it cannot bind to any RNA. So that's what we use. We did that, and the transcript, we did qPCR with the transcript, and then we found out that as we expected, the RAB4 mRNA is binding to TDP43, and the mutant is not. So this is genuinely an exciting result. So what we did to confirm this was to get human ALS patients. We got a high PLC derived motor neurons from actual human ALS patient carrying TDP43 mutations, and we repeated the same experiment, and it was very similar because the TDP43 mutant, the new motor neurons that were TDP43 mutant, they were not they were not binding TDP43. So this, can you see this? Okay, I can't take this away. So this is validating the result that we saw in Drosophila, that RAB4 is a direct target of TDP43. When I say RAB4, I mean messenger RNA. The mRNA of RAB4 is binding directly to TDP43, which says TDP43 is regulating RAB4 somehow, maybe translation, splicing, all these functions that TDP43 does. It has a physical relationship with RAB4, just like syntaxin and Futsch and all these other synaptic proteins. So this gives us a way to, we're running out of time. Okay. So this is this shows a conserved pathway across species for across species from Drosophila to human, that this RAB4 for TDP43 pathway is conserved. What is happening in the fly can also be seen in humans, so which gives us hope for a therapeutic implication. This is what I have said about Futsch. Everything that RAB4 did, Futsch 2 was able to do it independent of RAB4. So Futsch is doing its own rescue and RAB4 is doing its own rescue. But what connect both of them was TDP43. How? So we proposed that TDP43 loss, of course, that's the beginning. The flies don't have TDP43 from the nucleus, so there is a nuclear loss of TDP43. So we hypothesize, at least for the first time, that there is a connection between RAB4 and Forge in TDP43 ALS, because when TDP43 is lost, the RAB4 mRNA is downregulation. There is downregulation of RAB4, and this affects the recycling vesicles that impairs Futsch, that made it difficult for Futsch to be present for at the synaptic buttons where it needs to be translated and bind to the microtubules to stabilize the joint. And then this affects the process of endocytosis, exocytosis, that affects synaptic transmission, and the mutant have to do more work. Eventually, the motor neuron gets tired and die. So this is what happens. This is what we hypothesize happened to motor neuron before he died, before the motor neuron would die. So let's see. This the therapeutic implication and next steps. Well, we have we have we have okay, let's say we have a lot of pathways that we could check. We have shown quite a number of proof of concept that RAB4, at least the RAB4 way, we could go the RAB4 way, that synaptic vesicle recycling is important in the mechanism of ALS disease, and we could check some RAP4 modulators to see if we could get some compounds that will improve vesicle recycling and mRNA localization to the NMJ. Or we could go the MAP1B route, which is the Futsch route, by modulating proteins that would enhance synaptic growth, or we could go the way of both of them, like cancer. Cancer is pretty much like one compound cannot reverse the effect of cancer. So cancer is usually multi-drug. So perhaps ALS will also be that way, so that we can target RAB4 and RAB5 pathways together. That is the major therapeutic implication that we have proposed. This is a fantastic proof of concept. That is the basic research that has provided the proof of concept for therapeutic ALS research. This I have just listed the assays that we did, some of the methods. In case anyone is interested, I'm happy to explain what happened and how I did it. And I can tell you that it actually works because we did it. Some published methods actually don't work, because some of them we tried, it didn't work, but these ones actually worked because we did them correctly. So if anyone is interested, I can explain what happened and how to do it. And I know I've said a lot of stuff today, and maybe some people might be lost along the way, but these are the key messages that I want you to remember. TDP43 mislocalization is toxic, and it is the hallmark of ALS disease, and it disrupts synaptic organization. That's our hypothesis. So I have told you why Drosophila is one of the best models to use to model ALS, and it's because the mechanisms are conserved from RAB4 to Futsch. And the therapeutic direction we are looking at will be synaptic vesicle recycling, because it has brought together, it brings together TDP43, Futsch, and RAB4. So understanding why synapse fail may be the key to understanding ALS and developing early interventions that preserve neuronal function. So these are some of the references I've used. I used more than this, but these are the main ones. And I'd like to say thank you to ICGB Trieste, my boss Fabian, Julia, Rafa, and the others that make this whole story complete. There is still more, but I didn't want to overload us with a lot of stuff. This is just one story out of the many that we have. So thank you for listening. I will take some questions now, if there is any.
Thank you so much for such an insightful session. We'll take questions from the house and then take some online questions. Do we have questions from the faculty first, then the participants, and then we go to the online audience? Anybody? Comments or questions? Either one. All right.
Hello, Monsurat. This is Dr. Alex Whitworth. Thank you for your presentation. It was very nice. Can you hear me well enough?
Yes, I can hear you, Alex. Hi.
Can you hear me? Yeah. Okay, great. Hi. Thank you for that presentation. I think I missed the transition to how you identified why you started looking at RAB4. Was that identified through a screening process, or was that a good guess, or was that some information from elsewhere? It's a very interesting hit that you've got there. Maybe you might like to say something about how that can influence synaptic process, the RAB4 itself.
Okay. Do you want me to answer this, or okay, I'll take this first. Thank you, Alex. I think I didn't. Okay, it's majorly because we have I'll say it's a combination of both, because we know we know what these RAB GTPases were, And we have the inkling that because some other synaptic proteins that were involved in vesicle recycling were downregulated, and we wanted to check, connect what other synaptic proteins will be involved in all of this. And yeah, we've had this before. I think there was a screen done before in the lab that identified some RAB GTPases, and we just, in this current work, we just narrowed it down to RAB4 and RAB5. Those were the guesses, but we knew the RABs were involved before.
All right. Thank you so much.
Hello, Monsurat. How are you? I don't think you see me. Hello.
Ah,
How are you?
right there. How are you?
I'm good. Nice to see you again. You look great.
Thank you.
So I have a question following Alex a bit. I didn't understand the relationship between RAP4 vesicle trafficking defects and Futsch translation defects. Could you elaborate a bit more on that?
Okay. Okay, this is what happens. RAB4 is involved in this we call them recycling endosome, recycling vesicle, the fast one. Because in the flies, there is the fast one, there is the slow one, there is clathrin endocytosis, a bit of different pathways to achieve the same thing. But for RAB4 sort of binds to these fast recycling vesicles, those ones that are pretty fast, like the flash neurotransmission ones. So RAB4 is the one that's our hypothesis anyway, that RAB4 being regulated by TDP43, being binded to the TDP43. So we hypothesize that it's doing something, it's controlling it somehow. Futsch is it's a microtubule associated protein. So we hypothesize that Futsch is being released, released faster from the vesicles because RAB4 is being rescued. Do you understand the process? Because Futsch is needed at the NMJ to stimulate the synaptic growth during growth and Button's expansion and division. So RAB4
Oh, so you
makes
think you,
available the vesicles of Futsch to get there faster.
but why is Futsch in vesicles? I didn't know Futsch was in vesicles. Maybe that's what I'm missing.
Ah, let's
Yeah,
see.
that's what I'm confused. But we can discuss it. Yeah, we can give a chance to other participants and we can
Okay.
have a chat online. It's very interesting, though. Thank you very much.
Is the clustering. Okay, we can talk about it later, Isa, if you are still interested.
Hello, Monsurat. How are you?
Hi, Akaimo. How are you?
I'm very well, thank you. Very nice talk. Now, my question is, how faithful are you that by rescuing RAP4 function, you'll be recovering significant aspects of the disease, especially considering that you've got like 6,000 genes whose expression is changed in your mutant condition?
How what? Again, please.
So you mentioned at the beginning of your talk that in the mutant condition, there is like 6,000 genes that change their expression.
Yes.
Still, I find very surprising, that's what I love of your talk, that by putting back RAP4, you managed to rescue significant phenotype, which is the neuromuscular junction phenotype. So my question is, in the final event of a therapeutic application of your findings, just imagine that you find a drug that rescues somehow RAF4 function in the TPP mutant. So do you think that this will rescue the disease to a extent that the patient will
Okay.
benefit from this?
Okay. Thank you, Akaimo. What I would say is this work being a basic proof of concept, if you ask me, because we know, and I know that there is a lot more going on than that, but this is just a fraction of what has happened to the fly and what I have followed, what we have followed. I'm still sure that there is a lot more angles that we haven't followed. There are but some of these 6,000 proteins that we are talking about are not may not be direct target like that, may not be, how do I put it, to may not be in the direct pathway affecting this directly. So for RAB4, it was surprising as well because this is a synaptic vesicle protein. It was surprising that it was rescuing the phenotype of ALS without TDP43. So we were thinking that something is here that we could explore. So if we find a
So
molecule,
yeah,
I would say we should look at molecules that work with RAB4, not RAB4 alone, but some molecules that could mimic the
yeah.
action, that we could check the front and back before we jump to the level of it rescuing the phenotype in human.
So the reason I asked you this question is because perhaps when it's always a problem of when do you detect the disease and when do you act on the disease. So if the terminal phenotype is 6,000 genes have changed their pattern of expression or the levels, then it might be too late. That's why I was asking you. Probably the sooner that you detect the disease, the smaller the damage to the neurons.
Yes, you are right, which is what Fabian was thinking, that perhaps we need to take some step backwards, to see how this is happening, where this is coming from. I think we were thinking in the same line you had just now, that probably if we go back far enough, we could be lucky that the genes, the proteins that are involved are fewer that we could actually follow before it branch out to these
Thank you.
numerous proteins that it's impossible to follow all of them.
Thank you. Nice talking to you, Monsurat.
Thank you, Akaimo. It's nice that you're in Nigeria.
No. Okay. Hi.
Hi, Lola.
Hi, Monsurat. How are you? That was a great work.
Thank you
Congratulations.
so much.
I hope to see you soon.
Yes.
Thank you so much, Dr. Monsurat. We have just one question online from an online.
Okay, thank you, Dr. Monsurat, for the wonderful presentation. So I think my questions generally have been asked, but I think I have here that what other proteins, or were you able to find other proteins such as Futsch that were able to restore the RAB4?
Did you get that, ma?
Yes, I heard her, but
Okay.
yeah, let me see. Did we do something on RAB4 to see which RAB4 levels were increased? Yes, actually, because why we linked both RAB4 and and Futsch together was because when we take out when we express RAB, Futsch was recovered. When you express Futsch, RAB4 too was recovered, but not RAB5 in either way, in either direction. So it's kind of specific. Whatever is happening in there is specific to RAB4 and Futsch, not another GT base.
Okay, thank you so much, ma. So the question we have online, you mentioned that RAB5 is bound to late endosomes. Do you think the RAB5 mutant effect you observed could contribute to impaired aggregate clearance? And if so, through which mechanism?
Okay. The RAB5, yes, late endosome. Perhaps that's the mechanism it happens that the questionnaire has talked about. We really didn't check RAB5 further, other than the fact that it did not rescue our phenotype, and it was, let's say, we use RAB5 like a negative control, so we didn't really pursue RAB5 further. So perhaps that is the mechanism. I wouldn't be able to say further. We didn't check RAB5 further.
All right. Thank you so much for your time and for that wonderful presentation.
Oh, thank
Yeah.
you very
If we
much.
have any, yeah, thank you for joining us today. If the participants have any more questions, they can always reach out to you. And we just wanted to say thank you for taking your time to speak at the Fly Neuro-Can Workshop 2026. Have a great day, and have a great day, everyone.